Hello, good evening to all of you. Welcome to Unacademy. This is Anand Babu from Vijayawada, Andhra Pradesh. About myself, I have 20 years teaching experience in botany. I worked in a repeated organization and produced a number of medical students throughout Telangana and Andhra Pradesh. Today we are going to discuss about the very important MCQs on cell division. This is one of the very important topic. Usually the people get confusion here in attempting answers. So by understanding this concept, it will be easy for you to attempt any type of objective. So I selected this as it is very important for examination point of view. For all competitive examinations, NEET and other EAMCET competitive examinations, this is very important. Carefully listen. We will go through the bits one by one, and I will give clarification where you need it. Okay. And before that, one important point I am going to tell you was it here about subscription here. Take plus subscription. It is very affordable, and you can have plans for 12 months and 6 months. So it is very convenient, affordable price. Okay. And on my referral code Babu Live, you will get a 10% discount. This is a good platform to you. I feel to be proud in joining this academy. Why? Because I may develop the rapport with you, so that interaction giving me happiness, and definitely I will share my knowledge here. Okay, let us start now. Okay, our topic, that is cell division. Let us go to first. Here you see. Let us go to first bit, objective type here. And listen carefully, all of you. And if any doubt is there, leave your doubt in comment box. Okay, I will clarify. Okay, comment, and also how will it be. Okay, students, let us start with the first objective bit here. Okay. Here is a first objective type of question from the cell division here. Crossing over occurs between sister chromatids of the homologous chromosomes, 2 DNA strands of sister chromatid, 2 chromatids of same homologous chromosome, non-sister chromatids of the homologous chromosome. First of all, you have to understand crossing over occurs in meiosis. Prophase 1 of the meiosis 1. Okay, crossing over. Crossing over occurs, you see here, pocketing stage. Am I correct or not? Pocketing. Okay. Here, what you have to understand, crossing over, what is the meaning? You know very well. Crossing over occurs in pocketing subphase of the prophase 1 of the meiosis 1 here. Here, this is one chromosome. Okay, this is another chromosome here. Okay, crossing over takes place between the chromatids here. So this is chromatid, this is another chromatid here. So 2 chromatids belonging to 2 chromosomes. I mean, these 2 are non-sister chromatids. Okay, crossing over always takes place between non-sister chromatids. These are sister chromatids. Chromatids belonging to the same chromosome, they are sister. Crossing over will not happen between the same chromatids, and crossing over will happen between the non-sister chromatids which are belonging to two chromosomes. Okay, this crossing over is very, very important, you know very well. Recombinations. Am I correct or not? Recombinations are very important for execution of new characters in offsprings. So the right option here is 4th. Okay, is it correct? Non-sister chromatids of the homologous chromosomes means structurally, genetically both are similar. Okay, so this is the answer. Next, let us go to another objective type here. A cell with here 2n diploid number of chromosome 16 undergoes meiosis. Each of the 4 daughter nuclei formed at the end of the division will have. What is given? Just understand. Diploid number of chromosomes given here. Diploid number is given. Ultimately in meiosis, how many daughter nuclei are formed? 4 daughter nuclei are formed. Okay, each of the 4 daughter nuclei formed at the end of the division will have how many number of chromosomes? Here is in meiosis, first you have to understand. So this is diploid. Here number is given what? 16 is diploid. First division in meiosis now here. So here meiosis. Okay. How many here? Chromosome number is reduced to half here. 2 daughter cells will be formed. Each containing 8 cells. 8 reduced. Chromosome number is reduced in meiosis 1. Am I correct or not? Whereas in meiosis 2, what happens here? These 2 chromosomes here. And meiosis 2 here. In meiosis 2, chromosome number will not be changed. It is equal to mitosis only. How many number of chromosomes are there here? Here. So in 4 daughter cells also, in each cell 8 chromosomes are present. Why? Because here chromosome number will not be changed. In the meiosis 2. In meiosis 2, chromosome number is doubled. In meiosis 1, chromosome number is reduced to half. So daughter cells, each daughter cell will have 8 chromosomes. So right answer is what? 8 chromosomes. Okay. Next question. Next objective. Number of spindle apparatus. See here, this carefully. Listen what is given in the question here. Number of spindle apparatus. Spindle apparatus singular, plural. This is formed during the formation of 100 microspores from microspore mother cell. Okay, how many number of spindle apparatus are formed? Here, people here somewhat confused here. What we have to do? One point you have to understand. For 1 microspore mother cell, MMC, here on meiosis, how many here are formed? 4 microspores are formed. 4 micro. So how many microspores are given here? 100 microspores. So how many here? MMC, mother cells. 100 by 4 here. 100 by 4 means 25. How many spindle apparatus are formed here? Spindle apparatus means here, one point you have to remember. In meiosis here, happens now. Here meiosis, this is the first division, sorry. This is the first division here. And this is the second division, meiosis 2. Okay, but here this is one spindle apparatus. Here 1 spindle apparatus. Here 1 spindle apparatus. So 3 spindle apparatus. So 25 into 3. Okay, how many here? 25 into 3? 75. What is the answer here? B is the right answer. I mean 2 is the right answer. 75. So are you following, students? Number of spindle apparatus are given here. In meiosis, by meiosis 1 and meiosis 2, how many spindle apparatus required here? 3 spindle apparatus. That point you have to remember. In meiosis 1 and 2, here 3 spindle apparatus required. Here how many microspore mother cells here? 25. 25 into 3, 75. So 75 spindle apparatus are formed. Okay. Another question here. Number of meiotic divisions required to produce 100 seeds in angiospermy plants. So this is also a little bit okay, confusion here. Don't confuse. Here it is simple only, but you have to understand the concept first. Number of meiotic divisions required. Meiotic divisions given here. So here this meiotic division. To produce how many seeds here? 100 seeds. In an angiosperm plant. Seeds are formed by formation of male and female gametes only, no? After fertilization only seeds are formed, no? That point here. I will give you clarity here. Here is a, for the formation of female gametes. Here female gametes are formed, no? Okay? Female gametes. For the formation of female gametes, I mean in producing 100 seeds, meiotic divisions required here 100. 100 are required. For each, one female gamete is formed. For the formation of here male gametes here, in the formation of 100 seeds here, enough 100 is not required here. 100 by 4, 25. 25 meiotic divisions required only here. Here 100 meiosis are required for the formation of one female gamete. For the formation of one male gamete here, meiotic divisions required are 25. 25 plus 100. 100 plus 25. How many here? 125. So are you following? Are you following here? So 125 is the answer here. This is the right answer. Okay? So carefully listen here. Another bit here. How many chromosomes will the cell have at G1 after S and after M phase respectively? If it has 14 chromosomes at interphase. So I will give clarity. This is cell cycle. This is what is called cell cycle. Cell cycle is divided like this. Interphase studied by cell cycle. Here G1 and this is S. This is G2 and this is M phase. Am I correct or not? Mitotic phase. So what is given? 14 chromosomes are there in interphase. 14 chromosomes here are there. Here 14. Here how many chromosomes? You see here, tell me chromosome number. Yes. How many chromosomes? 14 only. In G2 also 14 chromosomes. But here what is the important point given? Okay. What is important? Phase respectively chromosome at interphase. How many? How many chromosomes will the cell have G1 after S, after M phase respectively? After M phase respectively. How many are there? 14 only. But one point you have to remember. In interphase, chromosome number will not be changed. Chromosome number will not be changed. Same chromosome. But what is changed? Chromatid number changed. Chromatid. Here you see. 2C to 4C. Here for example, here this is 2C, let it be, this becomes 4C. Chromatin only becomes doubled, 2C to 4C, but chromosome number will not be changed. Okay. In anaphase of the mitosis or meiosis, chromosome number will be changed. Chromosome number is not changed in interphase. So what is the right answer here? This only the right answer. 14, 14, 14. In G1, G2, I mean this gives idea of there is no change in the number of chromosomes in interphase. Okay. Next question. Here what is the number of cell divisions required to produce 256 cells from single cell? It's easy only. Okay. From single cell here 256 cells. How many number of divisions required? Okay. Easy. It is very easy. 1 to 2. Here 2 to 4, 4 to 8, 8 to 16 here, 16 to 32 and 64, 128 and 256 like this. How many? 1, 2, 3, 4, 5, 6, 7, 8. Okay. What is the right option here? 4 is right option. Okay. It is very simple. So these divisions we have to calculate like this. Okay. Let us go to another bit here, 7th bit. At what stage does the number of chromosomes become half? At what stage? In mitosis or meiosis? In any type of cell division, chromosome number will be changed only at one particular stage. Whether chromosome number is reduced or chromosome number is doubled. Only one is the phase, that is what is called anaphase 1. Anaphase 1. In anaphase 1, what happens? Chromosomes move to opposite ends. Chromosomes move to opposite ends. So like this. Okay? Chromosomes move. Movement of chromosomes happens. So during the movement of chromosomes, what happens? Number automatically will be changed. Okay? So anaphase 1 here given. Options, 4 options are given. Anaphase 1 is the correct answer. And 8th question. Let us go to come to 8th question here. Which brings about equal distribution of chromosomes? Equal distribution of chromosomes. In mitosis, you see equal distribution of, for example, here you see this is a diploid cell containing 46 number of chromosomes here. Here in mitosis, 2 cells are formed only, not 4 cells. Here the daughter cells also having 46. Equal number of chromosomes are maintained here. Here how it will be? How 46 equal number of chromosomes here? One phase here. For example, here the chromosomes are already told, before anaphase, at the anaphase stage only chromosome number is double here. Here you see 92 chromosomes here are there. Okay. Doubling of chromosomes takes place now here 92, 46 and 46 like this. 46 and here 46 like this. How, sir, 46 number of chromosomes will be found means only one point you have to know here. Centromere is there, no? This is centromere. The centromere splits like this. Okay, so 46 becomes 92. So 46 this side, 46 this side. Okay, that point. So equal distribution of chromosomes. So splitting of centromeres only. This acts as one chromosome, this acts as another chromosome. So which brings about the equal distribution of chromosomes means here splitting of centromere. Because of splitting of centromere, 2 chromatids get attached to centromere. Centromere splits. So what happens? Equal number of divisions happens. 92 chromosomes formed. These 92 chromosomes formed because of splitting of centromere. 92 chromosomes. 46 this side, 46 this side. So equal number is maintained here. Okay. And in meiosis, you see 9th objective here. In meiosis, a bivalent is an association of. What is a bivalent here? 4 chromatids are given. And 2 chromatids are given, 2 chromatids and 1 centromere. What is the right answer? Okay, bivalent, you see, bivalent means, you see this bivalent in zygote stage of the meiosis 1 of the prophase 1. Zygote. In zygote, chromosomes form pair like this. Chromosomes form pair like this. This is a pair. In this pair, how many chromatids are there? Tell me. 1, 2, 3, 4. 4 chromatids are there. 4 chromatids. How many centromeres are present? This is 1 centromere and this is another centromere. 2 centromeres. So what is the right answer? Tell me. 4 chromatids and 2 centromeres. 4th one is right. Bivalent is nothing but pair of chromosomes only. In a pair of chromosomes, how many chromatids are there? How many centromeres are there? These 2 chromatids, each chromosome consists of 2 chromatids, and like this 4 chromatids totally and 2 centromeres. Okay, this is the right answer. Okay, we'll switch over to another bit here. DNA molecules present in a bivalent is. In a bivalent again one word, same word, similar word is used. This is a bivalent. So bivalents occur in where? Zygote, which is the subphase of the prophase 1 of the meiosis 1. Here bivalent. Okay. How many DNA molecules are there? Very simple here. How many chromatids are there? 2 chromatids. Here this chromosome consists of 2 chromatids. And totally how many here? This is 1 DNA, 2nd DNA, this is 3rd DNA, and here another DNA. How many? 4 DNA are present here. 4. So in a bivalent or in 4 chromatids, 4 DNA molecules are present. Okay? Are you following? Are you following? Are you following? Okay. Another important bit here. Another important bit. Tetrad is made up of. What is the tetrad? Here the same here word. These are very, very important. They are very, very important here. Tetrad means. Here is a. Okay. Tetrad. What is the tetrad? Many people will get confusion here. What is tetrad? Some people say that 4 chromosomes together is called tetrad. It is incorrect. So this is one chromatid. This is another chromatid. This is another chromatid. This is another. I mean, in 2 chromosomes, how many chromatids are there? 4 chromatids are there. Okay. Tetrad is made up of 4 homologous chromosomes with 4 chromatids. 4 homologous. No, it is 2 chromosomes only. 2 homologous chromosomes, each with 2 chromatids. This is correct. Second option is correct. How many chromosomes are there in a tetrad? 2 chromosomes only. Chromatids number is 4. These 2 chromosomes are homologous or non-homologous? Homologous chromosomes. I mean, pairing occurs always in meiosis between homologous chromosomes. Okay, which are similar in structure and genetically also they are similar. So another bit here. What is the true of anaphase here? Read, chromosomes move to opposite poles. True. What is true? Not false. Chromosomes move to opposite poles. Spindle fibers move apart. Okay. Golgi bodies and ear are reformed. Okay. And anaphase, what is the right answer? Centromere split and chromatids separate. What is the right answer? Here it is very simple. And anaphase, this is not incorrect. This is incorrect. Third option is incorrect. Why? Because here Golgi bodies here are reformed in telophase. Spindle fibers move apart. Spindle fiber, this is not. Chromosomes move apart. This is also wrong. Chromosomes move to opposite poles. Opposite poles. Chromosomes move to opposite poles. How they can move here? That point here. Centromere split and chromatids separate. Okay. So in anaphase asked, chromosomes move. How these chromosomes are moving not given here. So this is also wrong. This is correct. Fourth option is correct. Centromere split and chromatids separate. Are you following? Okay. And here microtubule depolymerizing drug such as colchicine is expected to. Colchicine is one of the drug here. What is the influence of colchicine in mitosis? Okay, or cell division here. What is the right answer means? One point you have to understand. First option is correct. Inhibits spindle formation during mitosis. So during mitosis in prophase, no spindle fibers are formed, no, like this. The spindle fibers are very, very important to pull the chromosomes towards the opposite poles. Chromosomes become doubled or reduced due to these spindle fibers only. But when colchicine is applied, what happens here? Colchicine is given. This spindle fiber formation will not be there. So chromosomes do not move apart. They do not move to opposite poles. So what happens? Tell me, chromosome number will be increased. I mean, cell with more number of chromosomes will be formed. So inhibits spindle formation during mitosis, and which results the cell with more number of chromosomes. So abnormal cell is formed here. Okay, inhibit cytokines is not. It does not inhibit. Allow mitosis beyond. So these are not. This is the right option regarding the colchicine. Okay. Another bit here. Which one of the following is the best stage to observe the shape, size, and number of chromosomes in a cell? Okay. Which is the best stage? Okay, prophase, metaphase, anaphase, and telophase, like that. This is the best stage, metaphase. Metaphase. Physiology of the chromosome will be studied very well in metaphase only. In metaphase, how chromosomes will be, tell me? All chromosomes released into cytoplasm and arranged themselves in a particular row. just like a row, chromosome C. Equatorial plate chromosomes are present. Okay, this is the stage. The chromosomes can be visible very clearly. Shape, L shape, okay, J shape, I shape, okay, like these shapes. Okay, so the shapes can be observed very well in metaphase of the metaphase of the cell. I mean cell division. Another question here, 15th question. The stage during which separation of the paired homologous chromosomes begins is. Here chromosomes here, one point you have to understand. Chromosomes in pocketing, you see here, in zygote you see, in zygote here like this. Synaptonemal complex will be present. Between two chromosomes, synaptonemal complex is formed in zygote. Synaptonemal. Okay, this is a kind of ladder-like protein only. So this is very important to make the two chromosomes helping in alignment of the two chromosomes like this. I mean in zygote, pairing of chromosomes happens now. This pairing occurs synaptonemal complex, which is a ladder-like protein present between the two chromosomes here. So in one of the stages after crossing over, next what happens? Crossing over happens, recombinations occur in pocketing. Next stage of the zygote is pocketing. So after that, these chromosomes move apart. The chromosomes is here separate. Completely they will not be separated like this. So how can you say that these chromosomes moving apart here, distance between these two chromosomes will be more? Why? Because the synaptonemal complex, which is a ladder-like protein, starts to dissolve. So this is helping in separation of chromosomes like this. Okay. So by counting the distance here, calculating the distance between two chromosomes, we say that, and chromosomes are getting separated. But chromosomes will not separate completely. Still there will be chiasmata here, crossing over occurs here. Okay. In diplotin diakinase only complete separation will be taken place. And another bit here, 16th. The enzyme recombinase is required at which stage of meiosis? Recombinase enzyme. This is enzyme complex. Here one important point you have to understand. Pocketing stage, what is happening here? Crossing over takes place. Okay, crossing over takes place. Okay, two segments of chromatids exchanged. So for what is that, cutting of this chromatid and attaching it to another chromosome. Okay, this recombinase is very, very important enzyme. This is complex, involving cutting and attachment of the chromatids. Then only exchange of this genetic material, which means exchange of chromatids. Genetic information will be exchanged in the form of chromatids here. So which stage? Tell me here. Pocketing. This is the right answer. This is the right answer. Okay. Another important bit. 17th bit here, you see, during gamete formation, the enzyme recombinase participating during gamete formation. The enzyme recombinase participating during prophase 1 only, no? Meiosis 1 of the prophase 1, prophase 1 of the meiosis 1 is important here. Okay. And this is the answer. Okay, direct answer. Okay. Prophase 1 of the meiosis 1 is a lengthy process, including 5 substages. What are they? Leptotin, zygotin, pocketin, diplotin, diakinase. Okay. And another bit here. Chromosomes move to the spindle equator. So select the correct option with respect to mitosis. What is the correct option here? Chromosomes move to the spindle equator and get aligned along with the equatorial plate. Metaphase is right only. Chromatids separate, remain, it is wrong. Chromatids start moving towards opposite poles. No. Golgi complex endoplasmic reticulum will still as visible at the end of no. They disappear by the end of this. So this is the correct answer. 1. I mean chromosomes move to the spindle equator and get aligned along with the equator. Already I told you in metaphase. Whenever nuclear membrane in the prophase dissolves, the chromosomes freely released into the cytoplasm and they are arranged in the form of a particular row that looks like a plate. That is what is called equatorial plate. So chromosomes are arranged equatorially. Okay, this is the right answer. Another question: which of the following is key event of anaphase of the mitotic division? What is that? Related to this anaphase. Chromosomes move to spindle equator and get this is metaphase. Anaphase means centromere split and chromatids separate. This is the right answer. Already I explained, because of splitting of centromere only, chromatids will be separated. So this is the key event that occurs in anaphase of mitotic division. Okay, and which of the following is not a characteristic feature of the meiosis? See here, this A is correct. One point you have to understand, the meiosis, here is a meiosis here. Okay, replication of the meiosis 1 and meiosis 2. Replication occurs only once. That point you have to remember. Replication of DNA occurs only once. I mean, just before the meiosis 1, just before the meiosis 2, replication of DNA. I mean, synthesis of DNA will not happen. So, replication of DNA will happen only once. Very, very important point here. Here is a wrong option is given. You see, it involves 2 stages of DNA replication. One before meiosis 1, another before. This is incorrect statement. So, the asked question is also, which of the following is not a characteristic feature of meiosis? So, this is okay. So, time is sticking up. My referral code is here, Babu Live. So, on this referral code, you will get 10% discount. Okay, recommend your friends also to join in Unacademy. Okay, and follow the lesson properly and convey this. Why? Because this is a stage very important to you for the development of your progress. Okay. Thank you very much.